Hi everyone, we're going to be picking up where we left off on discussing meiosis. Okay, so something really important about meiosis, which is really our form of forming sex cells or gametes, is we want to increase genetic variation. So remember, meiosis only occurs in your sex cells, and the reason why we want to increase genetic variation is because you want your offspring to be as diverse as possible to increase the likelihood of survival for your species. So the question is, how does genetic variation occur? There are a couple of different ways. The first one we've briefly talked about is crossing over during prophase 1, where your two parent cells, your mother chromosome and your chromosome from your father, they will form a tetrad and cross over and exchange some DNA so that your resulting chromosomes will have a mixture of DNA from your father and from your mother on the same chromosome. So that's one way. Another way is called independent assortment, which occurs during metaphase 1. So what this means is that during metaphase 1, where your homologous pairs of chromosomes are lining up in the middle of your cell, it's possible that they're aligned perfectly, where all on one side is your father's set of chromosomes and the other side is your mother's set, and that would result with cells that are like completely like your father or cells that are completely like your mother, disregarding crossing over. But the second possibility, actually, when your chromosomes align in the middle of a cell, it can align in different ways. So it could be like 3 chromosomes from your mother's side and then 1 chromosome from your father's side and then 3 more from your mother's side. It can have just so many different variations and combinations of those, which side is mostly your mother's DNA and which side is mostly your father's DNA. So that random assortment is called independent assortment of chromosomes in the middle, and that just refers to the way that the homologous pairs of chromosomes align in the middle of the cell during metaphase 1. So that's another way you can have more randomness or more genetic variation in your offspring. The last one is random fertilization. So the book doesn't talk about this too much, but just know that the egg and sperm cell, the combinations of those are just pretty much almost infinite, the number of the different kinds of combinations you can have. And so that leads to also more genetic diversity in offspring. So there's a short section discussing homologous chromosomes, which but at this point you probably already know. But homologous chromosomes just means that on the same locus, which is same location, they both the homologous chromosomes code for the same characteristic. So for example, in this case, both chromosomes will code for eye color, but they will just be different ways you could do eye color. So brown eye color and blue eye color. Here, another one could be both coding for hair color. And so just notice that the characteristic is the same on homologous chromosomes, but just the actual manifestation of that is different. Okay, so just taking a more in-depth look at crossing over, which remember is one way to increase genetic diversity. Crossing over, it happens in prophase 1, and it's just you have 2 homologous chromosomes come together and form a tetrad. And as they form a tetrad, they form these things called chiasms. Chiasms are X-shaped regions where the DNA are like the chromosomes are crossing over, and they start to swap DNA. So every time these 2 homologous chromosomes touch each other, that's called a chiasm. So just to go in depth on how crossing over happens, first the homologous chromosomes will line up and make sure that all of their DNA is lined up accordingly. And after they line up their DNA, there's breaks that happen in the DNA in the same place. So in the mother chromosome, the break will happen in the same place where it happens in the father chromosome. And so once the DNA is broken, they immediately start to refuse with each other. So the DNA that's from your mother's side will fuse with the DNA from your father's side and vice versa. And you can see it in this picture. Here's the break that happens in the same location. And then you see how there's this crossing over at the chiasma where they refuse but with the opposite chromosome. And so this structure right here where there's kind of like all this crossing over, when the DNA gets separated during anaphase 1, then it will separate these chromosomes. So this allows for just more combinations. This process is called genetic recombination, and that's just a result of genetic exchange, which results again in different daughter cells from the parent cells. This last slide is about what occurs when meiosis does not go smoothly. We've been previously talking about how when meiosis goes as according to plan, all the chromosomes will evenly split. But sometimes that doesn't happen. There's something called non-disjunction, which is when the chromosomes don't split like they're supposed to. So one side will pull all the chromosomes to itself, and then the other side will lack chromosomes. So this can either happen at meiosis 1 or meiosis 2. So if it happens in meiosis 1, it's kind of more serious because it impacts all of the daughter cells. In meiosis 1, the homologous pairs of chromosomes will not separate. And so what you'll have is the two daughter cells after meiosis 1 will have, 3 sets of chromosomes, and the other one will only have 1 set of chromosome, and that affects the eventual daughter cells of meiosis 2 in that process, because you'll have 2 daughter cells that have 1 extra chromosome than it's supposed to, and then 2 daughter cells that will lack chromosomes that it's supposed to have. So non-disjunction in meiosis 1 will affect all of the daughter cells. You can also have non-disjunction in meiosis 2. So that means that the homologous pairs of chromosomes separated like they were supposed to, but then in one of the daughter cells, the pair of sister chromatids will not separate. And so in one of the daughter cells will separate just fine, and you'll have 2 of the daughter cells being exactly like what they're supposed to be. But if the sister chromatids do not separate, then one of the daughter cells will have too many chromosomes, and the other one will lack 1. So these are just 2 different forms of non-disjunction. In meiosis 1, the homologous pairs of chromosomes don't separate, and in meiosis 2, the sister chromatids do not separate. So these are just 2 ways that you can alter the chromosome numbers in your eventual daughter cells, and all of these would have serious implications for your offspring. Okay, thanks so much for watching. Bye!